Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Today in practical number 6, we will keep going to detecting for carbohydrates through a group of tests. Today we are going to talk about two tests: osazones formation and iodine test. Okay, we will start with osazone formation test. First of all, let us to know what osazone is. Osazone or osazones, because it's a group of chemical compounds. They are a carbohydrate derivative generated when reducing sugars react with the excess phenylhydrazine. This phenylhydrazine is a compound. It's a common reagent for the osazone test. So when this phenylhydrazine react with the aldose or ketosis or aldoses sugars, which are reducing sugars, so at high temperatures, so the osazones will formed as a result of a positive reaction. So this reaction, we can distinguish between reducing and non-reducing sugar because it's basis on the reaction between phenylhydrazine with the carbonyl group at reducing sugars. If there is no free aldehyde or ketone group, so this sugar is non-reducing sugar. That means the result will be negative. And also we can distinguish between the non-reducing sugars also, because the osazones form as a crystals. These crystals have different shapes, different structures, and different. So the crystals have different shapes and different time to formation. So the distinguishing between the non-reducing sugars, or the distinguish between the different non-reducing or the different reducing sugars will be based on the structure of the crystals and also the timing of formation of these crystals. The osazone test. It's a chemical test for detecting reducing sugars. This test can even distinguish between the reducing sugars on the basis of how fast a complex can be formed. That means it depends on the time of formation of the crystals. So how fast a complex can be formed. Because of the reagent employed, or the common reagent in the test, this test is also known as the phenylhydrazine test, because the common reagent in the test is a phenylhydrazine compound. So the test also known as phenylhydrazine test. So you can hear osazone test or phenylhydrazine test. The objectives of the test to identify sugars that are reducing in nature, to distinguish between reducing and non-reducing sugars, as I said to you. If the sugar is not in non-reducing sugar, so the result will be negative. to discriminate or distinguish between different types of reducing sugars.
هي قفل الباب.
To distinguish between different types of reducing sugars, as I said to you, because different sugars or different reducing sugars have a different time for formation of ozones, and also had different shapes and structures for ozone crystals, because the result will be as a formation of crystals, not a color. The final result of the test will be a formation of ozone crystals. Okay, so the requirements to do the test: first, the reagent. To prepare the reagent or ozone mixture, add 0.5 gram of phenylhydrazine hydrochloride and 0.1 gram of sodium acetate. So this is the reagent: phenylhydrazine hydrochloride and sodium acetate. And also the other materials and chemicals, the glacial acetic acid and test sample. This is a sample, we will test it. And the materials required are test tube stand and pipettes, and also equipment are vortex. As you know, it's mixture, okay, and water bath and microscope. This microscope, we will need it to see the crystals and the formation of different crystals for different reducing sugars. The principle of the test is based on a simple principle. This test uses phenylhydrazine in acetate buffer as a reagent. The basis of the ozone test is that phenylhydrazine reacts with the carbonyl group, which should be free. In reducing sugars, the carbonyl group almost will be free, so they can reduct and oxidize another compounds. Because of that, they are reducing sugars. If there is no free carbonyl group in the sugar, so that means it's a non-reducing sugar. Okay, so this test, the principle of it is that the phenylhydrazine reacts with the carbonyl group, which should be free in the reducing sugars of the carbohydrate to form ozone. Okay, so this given below is a reaction showing glucosazone, because it comes from glucose, so it will be glucosazone formation when D-glucose interacts with phenylhydrazine. So if this sugar is fructose, will be the yield will be fructosazone, and if it's lactose, this yield will be lactosazone, and so on. Okay, because it's a carbohydrate derivative. It comes from carbohydrates when they react with the phenylhydrazine. So this is the carbonyl group, and it's free in the reducing sugars, as I said to you. In this situation, the carbonyl group is it's on the anomeric carbon, or the carbon 1, right? So this is aldose sugar. But if it's a ketone sugar, the carbonyl group will be at the carbon 2. Okay, so the reaction will interact with phenylhydrazine will be at these two carbons, carbon 1, which contain a carbonyl group, and also carbon 2, as you see in the result. These two carbons, 1 and 2, and the 3, 4, 5, and 6, they stayed without any changing. Just the reaction involves these two carbons, 1 and 2. Okay, oxazone crystals are yellow colored and have a certain solubility and shape, melting point, and formation time. That means each sugar have or has own oxazone crystals comes from the interacts with a phenylhydrazine will be has own solubility and shape and melting point and formation time, which are differ from the another sugar. Okay, so through that we will distinguish between the different reducing sugars. We will say these crystals for glucose, or these crystals for lactose, or these crystals for maltose, and so on, according to the shape of the oxazone crystals and melting point and formation time, because they are differs from sugar to another. Okay, various sugars require different amounts of oxazone. Various sugars require different amounts of oxazone. C2 or carbon 2 epimers yield the same oxazone as carbon 1 epimers. That means if the carbonyl group at the carbon 1, and also if it's at carbon 2, the yield will be the same oxazone, because the reaction involves the two carbons, carbon number 1 and carbon number 2. So if the carbonyl group is on the carbon number 2, or it was on the carbon number 1, the yield will be the same oxazone. Okay, because both carbon 1 and carbon 2 are engaged, or they are involved in the reaction, so they will give the same result of oxazone crystals. Ketones having the same structure as aldoses below C2 or carbon 2 produce the same oxazone. Why? Because the the another carbons in the sugar below the C2, like carbon 3, carbon 4, carbon 5, and 6, they all have the same structure between ketosis and aldosis. Okay? Like fructose and glucose. The carbon structure under carbon number 2 produce the same oxazones. Ketones having the same structure as aldoses. Why? Because carbon number 2, or because below the carbon number 2 they are the same, so they will give the same oxazones. I hope you understand this. I will draw it for you to show you. This is the sugar structure, or wait, wait a moment. So we said the ketosis has same structure as aldoses below the carbon 2. So this one carbon 1 and this is carbon 2. Okay, so below carbon 2 the aldoses and ketosis having a same structure. Okay, below carbon 2 they have a same structure, so the oxazole reaction involved these two carbons 1 and 2. So because the same structure for the other carbons in the ketosis and also in the aldoses, so they produce the same oxazone crystals. Okay, okay, the procedure: take 5 ml of the solution under the test or the test sample should be placed in a dry, clean test tube, and add glacial acetic acid 5 drops and the oxazone mixture, which we prepared it already. Take from the mixture 0.3 gram to the test tube. So we add 5 ml from the sample solution and we adding 5 drops of glacial acetic acid and also 0.3 grams of oxazone mixture. Then mix everything up thoroughly, and if required, gently heat the test tube in a water bath to help the components dissolve. Okay? Watch the crystals form at various time. At various time, that means the crystals will appear or will form as or on a different time for a different sugars, reducing sugars. So if the sugar in the sample is non-reducing sugar, there is no crystals will be formed. Okay, but we are talking in a situation of this sample having a reducing sugar. Okay, so after heating on the water bath, heating the test tube on the water bath to help the components dissolve, so watch the crystals form at various times and try to record the time while keeping the test tube submerged in the hot water bath. Use a modest magnification in a microscope to look at the crystal's shape. So this is the procedure in the picture. So this is a solution of a sample. We dissolve a carbohydrate in water and adding 5 drops of glacial acetic acid and 0.3 gram of the ozone mixture or phenylhydrazine reagent, and gently heating on the water bath. And you can observe on the various time, you can observe the crystals started to formation or to form. This is the crystals at the end of the test. So that means the carbohydrate of these samples contain a reducing sugar, and from the shape of the crystal and also from the timing of formation, we can determine what this type of sugar, or we can identify what this type of sugar from both the formation of the crystal and the timing of the formation, the crystal structure or the crystal shape and the timing of formation. Okay. Results and interpretation. The structure, the form and appearance time of the crystals can be used to distinguish between various sugars, as I explained this for you. The following chart shows how to identify reducing sugar using this test. So here we have the sugars. They're all are reducing sugars. Okay, lactose, maltose, galactose, glucose, and fructose. And this is the crystal's appearance time per minute. Okay, and this is the structure of crystals. So from the structure and the formation or appearance time of the crystal, we can distinguish between them, because as you see, they have a different time of appearance, and also they have different shapes. For example, lactose crystals, lactosome will appear at 30 to 45 minutes, and also maltose will appear at the same time, 30 to 45 minutes, but lactose and maltose have different shape of crystals. The lactose crystals shaped as cotton ball or powder puff shape, but maltose crystal structure like sunflower or star shape. These shapes, you can see them under microscope. Okay. For galactose, the crystals appear at 20 minutes. The shape of crystals, thorny ball shape. Glucose appear at 5 minutes. The structure of crystals like needle shape. And the fructose the crystals appear at 2 minutes, and the shape of crystals like needle shape also, as I said to you, because they are same as the structures below carbon number 2, so they give same OSA zones, needle shape. Okay, so this is the shapes under microscope. This is the shape of OSA zone crystals. As I said to you, this test result basis on the appearance of the crystals, not the color of the final solution as the other tests or other previous tests. This test based on or the result of the test based on the appearance of the crystals and the time of the appearance. Okay, so as you see, the different sugars have different crystals structures. As you know, this is lactosazone, which comes from lactose with the reagent. Okay, the crystals like the cotton ball shape, and the maltose with the reagent of ozone hydrazine give a maltosazone. The crystals like sunflower shape, and the glucosazone comes from glucose with the hydrazine, phenyl hydrazine reagent. The crystals shaped like a needle shape, and so on. You can see the different shapes of different reducing sugars: lactose, xylose, maltose, mannose. You all have different shapes of the ozone crystals and also different formation time. I hope this is all clear, and we'll go into the test number 2, which is iodine test. Okay, before we shift to the test number 2, this is for picture for lactosazone crystals. It's shaped like the cotton ball. Okay, this is a clear picture. You can see them under microscope. In the applications of ozone, the ozone test is the sole means to distinguish between lactose and maltose when identifying unknown sugars, because they have a different crystal shapes, maltose and lactose. This is a straightforward test for detecting and differentiating the many sugars used in clinical practice that is also less expensive and time consuming. This test can also detect sugars in plant tissues. So, despite this test is useful, but it has some limitations. Like, so sucrose is a non-reducing sugar. It should give a negative result, right? But when it boiled for longer than 30 minutes, this test yields as a positive result for it. So if you boil the sucrose while it's a non-reducing sugar, but if you boil it more or longer than 30 minutes, it will give a positive result. Also, this test will not be reliable of the sample being tested contains a blend of sugars. That means if the test sample containing a mix of sugars or blend of sugars, the test will not be reliable. Additionally, a lot of sugar is necessary for a successful conclusion. Okay, this is about ozone formation test. Now we are going to talk about iodine test. Iodine test is a chemical test used to distinguish mono and or disaccharides from certain polysaccharides. The polysaccharides like amylose, dextrine, and glycogen. This test has a variation termed starch iodine test. It's also iodine test, but to detecting the starch. So it's termed starch iodine test, that is performed to indicate the presence of glucose made by plants in the leaves. So, as you know, the starch is the storage form of glucose in the plants. The plants store glucose in their tissues as starch. Okay, so if you want to detect the presence of glucose made by plants, so you can do this test, which terms starch iodine test. So, as you know, the polysaccharides are long chain polymers of monosaccharides. They are connected to each other with a glycosidic bonds. Okay, for example, this is a starch, consists of amylose and amylopectin, which is a branch section, and the amylose is a linear structure without branching. And this is a glycogen, and the glycogen is a form of glucose storage in the human and animals. Okay, it's storage in the liver. The glucose is storage in the liver as a form of glycogen. But in the plant, the glucose storage as a starch. Okay, and we have also the cellulose. It's also a polysaccharide, but it has a different shape of the monosaccharides polymers. The objectives of the iodine test to detect the presence of polysaccharides, primarily the starch. Okay, so we will detect the presence of the polysaccharides and primarily the starch. Principle of iodine test. The iodine test is based on the fact that polyiodide ions form colored adsorption complex. That means this complex can absorb the color. Okay, so the polyiodide ions form colored adsorption complex with helical chains of glucose, residue of amylose, and will give a blue-black color. And if the sample was a dextrine, it will give a black color, and glycogen will give a reddish brown color. Okay, they are polysaccharides, but each one have a different color due to the helical chains. Okay, so the monosaccharides and disaccharides and branched polysaccharides like cellulose, as you know the structure of the cellulose, these four sugars or these four saccharides remain colorless because they cannot do the helical chains. Okay, the amylopectin produces an orange-yellow color. Amylopectin, it's branched but not like cellulose, so it gives or it produces an orange-yellow color. The reagent used in the iodine test is Lugol iodine, which is an aqueous solution of elemental iodine and potassium iodide. So the principle of the test based on the polyiodide ions, which comes from the reaction between the elemental iodine and the potassium iodide to give a polyiodide ions, and then this slipped in the helical chains of the polysaccharide and absorb the light to give the certain color. Okay, but at first the reaction will be with the elemental iodine and potassium iodide at first, and then with the helical chains of the polysaccharides. Iodine on its own is insoluble in water. The iodine on its own is insoluble in water, but when it's or addition of potassium iodine, or when it's with potassium iodine, it will be dissolved in water. Okay, so addition of potassium iodine results in a reversible reaction of the iodine ion with iodine to form a triiodide ion, as you see the reaction below. This is potassium iodine. Okay, so the reaction will with the potassium, or addition of the potassium iodide to the iodine or elemental iodine, will give a reversible reaction. This is the aqueous solution. Okay, so then the iodine from the potassium iodine will react with the iodine ion to give a triiodide, and then also will react again with the iodine ion, this component or this compound triiodide, we react with another iodine ion to give pentose iodide, and then this is the polyiodide ions we are talk about. Okay, so which further I will repeat again. Iodine on its own is insoluble in water, as I said to you. So addition of potassium iodine results in a reversible reaction like this one of the iodine ion with the iodine to form a triiodide. Okay, which these compounds will further react with an iodine molecule to form a pentoiodine ion, and this is the polyiodide ions we are talking about. Bench iodine solution appears brown. The solution of iodine appears brown, whereas the iodide and triiodide and pentiodide ions are colorless. Okay, it's observed that the helix structure of the glucose chain is key to the test. So the polyiodide, which is triiodide or even pentiodide, they are colorless, but when they are slipped on the helix or when they are bent to the helix structure of the polysaccharide or the helix of the starch, when the polyiodide bent to the helix structure, they will give a certain color, like blue for the starch, like black for glycogen, and so on. Okay, but as their own, they are colorless. The pentiodide and triiodide and the iodide also, they are colorless. But when they bind to the helix structure of the starch or of the polysaccharide, they will make a color absorption complex. Okay, further, the resulting color depends on the length of glucose chains. So if the glucose chain was longer, so the color will be in high intensity. If the glucose chains was longer, so the color will be in high intensity. So the resulting color depends on the length of the glucose chains. The triiodide and pentiodide ions formed are linear. Okay, they formed linear and they slip inside the helix structure. I will draw it for you. The ions, or let us at first to draw the linear or the helix structure. Let us to imagine this, the helix structure, and if there is another color, it's purple. So the polyiodide ions formed linear, okay, not branched. So they slipped inside the helix structure. If it's okay, they are inside the helix structure. Wait. To appear like it's inside the helix structure. Okay, so this is the polyiodide ions, and it forms linear, and it slipped inside the helix structure, and so this is the complex which is absorb the color. Okay, it's believed that the transfer of charge between the helix and the polyiodide ions result in changes in the spacing of the energy, because the ions have a negative charge and the helix have a positive charge. So the transfer of charge between both of these helix and iodide ions results in a change in the spacing of energy levels, which can absorb visible light. So this transformation or transformation between the two charges of polyiodide and the helix can absorb the visible light, okay, and given the complex its certain color. Okay, the intensity of the color decreases with the increase in temperature. This is as a factors, you can say this is a factors affect on the appearance of the color or appearance of this complex, which are the temperature, number one. So how the temperature can affect through it can decrease the color. So the color will not form if the high temperature. In high temperature, the color will not form, or on heating, the colored polysaccharide iodine complex, which is this one, this complex, dissociates. On heating, this complex with iodide, polyiodide and the helix structure, they will dissociate or disconnect on heating. Because of that, the color will decrease. Okay, but on cooling, the color will increase again, because they will or due to the recovery of iodine binding capacity, and due to regaining of the helix structure. So they will bind again on cooling and give a certain color. But in high temperature or in heating, the color will decreases. And also another factor, the presence of water miscible organic compounds. Water miscible organic compounds like ethanol, they also affect as they dissociate the complex, this complex, the iodide and the helix will dissociate again in the presence of water miscible organic compounds like ethanol. Okay, on heating, the blue color amylose iodine complex dissociates. As I said, it's just a repeating for the explanation. The blue color on heating, the blue color amylose iodine complex dissociates but is formed again on cooling because the helical structure is disrupted. Thereby, amylose loses the iodine binding capacity and the blue color. Okay, the helical structure on heating also can affect because it's disrupted or the amylose loses its iodine binding capacity. It can bind to the helix or to the iodine. The amylose loses its iodine binding capacity on heating, yes. And also the color also will decrease. This is on heating, but on cooling the color will form again on cooling. Okay, the blue color reappears on cooling due to the recovery of iodine binding capacity due to regaining of the helical structure. Okay, I just try to form a model as a laboratory to do the or to explain the test procedure. Here we have two test tubes, okay, having certain samples. We will test them if they're containing starch. So we will add a few drops of iodine solution, which consists of iodine and potassium iodine. Here we adding the drops, okay, and then try to observe the color change. So if the color change, so that means the sample containing starch, and if the color remaining without changing, so it will be a negative result and the sample do not containing a starch. So this one changed in color, given a blue color, detect or indicate that a sample containing a starch, and this is a positive result and this is a negative result. But if the sample given another color like black or orange yellow color, so that's mean the sample containing a polysaccharide, because as you know, the different polysaccharide has a different color for the complex between helix and the polyiodide. It due to their difference or their variation on the glucose chain and the lengths of the glucose chain. Okay, but cellulose give a negative result. because it can do a helix structure. So why does iodine turn starch blue? This is a helix structure, which I had drawn to you. It is a helix structure. It's a polymers of glucose chains. Okay, it is called a helix structure, and this is the linear of polyiodide with a purple color. They are marked as a purple color, but on the nature they are colorless. Okay, but when they slipped on or inside the helix structure as in the picture, to give this complex called starch iodine complex, they will give a blue color because of this complex can absorb the visible light to give a certain blue color. Okay, that happens due to the transfer of charge between the helix and the iodine. The iodine can bond in amylose structure of starch, then negative charge in iodine transfer to each other. Okay, this reaction causes energy absorb the light and exposed in deep blue color to our eyes. Okay, so the requirements to do the test: first, the reagent, the Lugol's iodine. We prepare it with 5% elemental iodine is mixed with 10% potassium iodine to form the Lugol's iodine. This is the reagent. Okay, then we have a test sample. The materials required are test tube stand, and the equipment is water bath. The procedure of iodine test: take 1 mol of a given sample in a clean dry test tube. Take control of 1 mol of distilled water in another tube. So this tube will mark as blank with no sample, just a distilled water. Add about 2 to 3 drops of Lugol's solution to both the tubes. 2 to 3 drops of the Lugol's solution to both the tubes and mix them in a vortex. Observe the appearance of color in the test tubes. Heat the test tubes in the water bath until the color disappears. That to experiment that the color will be decreased on heating, as I told you. So in heating you will observe the color will disappear after its appearance before heating. So in heating you will observe the color will disappear, and then we will cool them again. Take the test tubes out for cooling. Note down the appearance of color. Sorry for interruption. So after or on heating, you can observe the test tubes will lose their colors, or the samples will lose their color. You can observe the color disappears, and then we will take them to cooling. Take the test tubes out for cooling. Note down the appearance of color seen in the test tubes. So the color will appear again on cooling. Okay, so you must note down which color will appear. It depends on the type of polysaccharide, if it's starch or if it's glycogen or even dextrine or so on. Okay, but cellulose will give a negative result, so there is no color will appear. The result and interpretation of iodine test. This is brown or yellow color. This is negative test for starch. That means there is no starch in this test tube, because in this situation they are testing for starch. Okay, so in the general iodine test, you will say the negative test will be on the no changing in color in the test tube. With no changing in color on the test tube means the negative result. So that means no polysaccharide in the sample, because the yellow or brown colors, they are indicated for a polysaccharide, right? We said amylopectin give a yellow orange color, and I think glycogen give a brown color. So in this situation, because they are detecting for the presence of starch primarily, so this will be a negative result. That means the starch is absent in this sample. But this sample, you can see the blue purple color, so it's a positive test because the starch is present. Okay, the appearance of blue, black, purple color represents a positive test indicating the presence of starch, a blue, black or purple color. If there is no change in color, the result is negative and indicates the absence of starch and even a polysaccharide. If there is no change in color. But if there is another color appear, like brown, yellow, orange, black, that indicate for another polysaccharide like glycogen, amylopectin, and so on. Okay, uses of iodine test. This test is used to detect the presence of starch in various samples. Similarly, this test is performed to test the process of photosynthesis in plants. As I said at the beginning of this test, we can detect for starch or the glucose made by plants in their leaves. So it's in indirect, you can detect for the process of photosynthesis in the plants. The limitations of iodine test. this test cannot be performed under acidic conditions. Also, as the factors of all the factors which is affect the test or the iodine test, we said the heating, and also we said the water miscible. Also, the acidic conditions also can affect the iodine test, because the starch and polysaccharides will hydrolyze, and in these acidic conditions and so on, the complex between the iodine and the helix structure will not formed. So this test cannot be formed under acidic conditions or at the lower pH. Okay, because the starch hydrolysis under such circumstances. This test is a qualitative test, just to detect if there is a starch or not, if there is a glycogen or not, but does not signify the concentration of starch. You cannot determine the concentration of starch or even the certain polysaccharide. Just you will detect if they're present or not on the sample. Okay, because it's a qualitative test. I think this is all about the iodine test. And if there is any question, you can ask me. Okay, see you later, and bye bye.